continue our discussion on carbonyl-containing functional groups, and here we're going to look at esters and amides. These are functional groups that look similar to carboxylic acids, but they have small changes. So the ester has the general structure RCOOR prime, with the oxygen atom single bonded to a carbonyl group and an alkyl group. So effectively this looks like a carboxylic acid, except your hydrogen has been replaced by an alkyl group. Of course, the carbonyl section is planar, and then the OR group can freely rotate and has a three-dimensional structure. So the simplest ester that you could have would be R and R prime, R being a hydrogen, R prime being a methyl group, and that's methyl methanoate. Naming esters, we have two pieces. The substituent name has to be there for the R prime, that is the part that's replaced to the hydrogen, and then the parent name for the chain of the carbon atoms that contains the carbonyl carbon. The systematic ester names have the pattern alkyl alkanoate, where alkyl is the R prime and the alkanoate is your main chain. There are common names for some esters, and they're in your text if you want to look those over. We won't focus on those. So the common name for propyl ethanoate is propyl acetate. So I think ethanoate going to acetate or methanoate going to formate would be your kind of your primary modifications. But let's look at a couple of quick examples here. You can see on A you've got a propyl group attached to your carbonyl, so that looks like a propanoic acid. But of course it's not the acid. You've replaced the proton with a methyl group. So we would call that R prime, that's the alkyl part. We would call this methyl propanoate. Next up in B, then you can see we've got CH3COO sort of system here. That's ethanoate. It looks like, kind of looks like acetic acid or the acetate. We call this ethanoate systematically. The hydrogen here in this case has been replaced by a propyl group, so we will call this propyl ethanoate. That is the IUPAC or systematic name. We can see the relationship here structurally between the various forms. We have the carboxylic acid form first, its conjugate base, the carboxylate ion, and then the corresponding ester, where we've attached an alkyl group where the hydrogen was. The ester functional group has polar carbon-oxygen bonds, so clearly it's a dispersion force-driven system, and so it exhibits dipole-dipole interactions which are quite strong. And as the R group gets bigger, the strength of the attraction between the esters goes up, but it's really due to the overall size, the dispersion force effect. So as a general rule, though, the boiling points and the melting points of esters are much larger than the corresponding alkanes, as you would expect. Now, esters lack an OH bond, so they can't act as hydrogen bond donors, but they can act as hydrogen bond acceptors through both the carbonyl oxygen bond or the in-chain oxygen bond, or oxygen atom. Now the next functional group we'll look at is amides. And the amide looks like a carboxylic acid, but you replaced the OH part with something that looks kind of like an amine. But of course, since the nitrogen has to bond to the carbon, you've only got two alkyl groups attached to the nitrogen. So amides have a general structure RCON, R prime, R double prime, and R prime, and R double prime can be either a hydrogen atom or a hydrocarbon. And here is the simplest amide, shown here with the oxygen in red, the carbonyl carbon in black, the nitrogen in blue, and then the other remaining 3 atoms are hydrogens, and this is called methanamide. Now we have to understand here there can be multiple alkyl groups, so this is going to be more complicated for naming these systems. So that leads us to classifying the systems. They can be primary, secondary, or tertiary, and that depends on the number of carbon atoms that are bonded to the amide here. So for the primary system, you can see you've got NH2 bonded to the carbonyl carbon in the amide. That's the simple amide. If you take one of the hydrogens on the nitrogen and replace it with an alkyl, you have a secondary amide. If you replace both hydrogens with an alkyl group, we have a tertiary amide. So this is primary, secondary, and tertiary amides. Naming primary amides is fairly straightforward. We take the alkane name, we replace the final E, and add amide. So you can see here are 2 simple examples. We've got the 2 carbon system, and then double bond to O, NH2. That's going to be ethanamide. And here's the C3 system. That's propanamide. There are common names, and you can deduce these in parallel to what we've already studied. Secondary and tertiary amides have to be named such that we identify the alkyl groups on the nitrogen. So we use the N prefix. It's similar to what we've seen earlier. The N prefix on the R group indicates their location, and then we alphabetically place them onto the name. So here we have 2 secondary amides. So we have an ethanamide base in A, because it's a C2 system, and then we've got the nitrogen with 1 alkyl group, and that's a propyl group, so we would call that N-propyl ethanamide. Next up, we have the propyl group on the carbonyl and a methyl group on the nitrogen, so we would call this N-methyl propanamide. The tertiary systems, we have 2 nitrogen groups, and if they're different, like in C, then we could have, for example, a methyl and a propyl, so we would call this N-methyl and propyl ethanamide. Or if they're both the same, like in D, we can have NN-dimethyl propanamide. The physical properties for amides, we can deduce that we have a very polar carbonyl group and a slightly polar carbon nitrogen group. So of course we've got dipole-dipole interactions on top of our dispersion forces. The primary and secondary amides have at least 1 NH bond, and these can act as hydrogen bond donors. Now all the amides can be hydrogen bond acceptors because of the carbonyl group and the nitrogen that will be present. So they all exhibit dispersion forces and hydrogen bonding forces at varying levels. Now, with a given structure, boiling point always goes up as size goes up, as it has for all the systems we've looked at in this course. So small R groups are very soluble in water. They're basically miscible in water. For larger R groups, solubility tends to decrease in water. The boiling points can be seen here. The primary and secondary amides, they can donate as well as accept hydrogen bonds, so they have higher boiling points than do the tertiary amides. Tertiary amides, in their pure form, don't have hydrogen bonding. They only have dipolar forces. So you can see here's comparing three different amides, a primary, a secondary, and a tertiary amide. They're all the same molar mass. We've just rearranged the atoms. The tertiary amide has two methyl groups, and that has the lowest boiling point. There's no hydrogen bonding occurring here. The secondary amide has nitrogen-hydrogen, which can act as a proton donor, hydrogen bond donor, as well as the carbonyl and nitrogen acceptors. So this has a much higher boiling point. See, it's almost 50, it's over 50 degrees higher. And then the primary amide has two hydrogens. It has a higher level of hydrogen bonding because there's multiple hydrogens, hydrogen bonding here. And so again, the boiling point goes up a little bit further. Now let's look at the reactions, common reactions that occur with these systems. We're going to look at what we call condensation and hydrolysis reactions. These are very important reactions. They're involved in protein formation and other biological system formation, in formation of polymers and other areas. So first off, let's look at what's called esterification. Esterification is a condensation reaction. And so in a condensation reaction, two organic molecules form a covalent bond and they release some small molecule. Typically it's water. Water is very thermodynamically stable, and that tends to be what we call the thermodynamic driving force, the energetic driving force for the process. So when a carboxylic acid comes in contact with an alcohol, so these are both dry systems, a dry carboxylic acid and a dry alcohol. Potentially this could be done in a hydrocarbon solvent. If this is done with heat, and very often acid acts as a catalyst, you'll see water is split out. The proton on the alcohol and the hydroxyl on the carboxylic acid combine to form water, and then RCOOR forms. We produce an ester. So in laboratory settings, if we're going to do an esterification reaction, we typically require an acid catalyst and heat. In biological systems, there can be biological catalysts that can control the growth of these systems. Correspondingly, we have esterification, we have an amidation reaction. Amidation is a condensation reaction where carboxylic acid and a primary amine, secondary amine, or ammonia react to produce the amide with, again, water being released. So typically in a lab, when we do this, we carry out this with heat and coupling agents. So tertiary amines lack an NH bond and can't undergo amidation reactions, only primary and secondary amines. So here, let's look at, in this figure, we're going to have a secondary amine. So we got maybe R prime and R double prime are alkyl groups. So we have a nitrogen-hydrogen bond that can react with the carboxylic acid. Now you might think, well, this could be an acid-base reaction, so we have to avoid those conditions typically. So we carry these out into conditions where the acid-base reaction is not favored, but the dehydration or amidation reaction is favored, where H2O is lost and the amide is produced. So we can do the reverse of this, and this is hydrolysis of esters and amines. And the esterification and amidation are reversible. The reverse is hydrolysis. So hydrolysis of an ester then will break the CO single bond and produce a carboxylic acid and an alcohol. And the hydrolysis of an amide breaks the carbon-nitrogen bond to give a carboxylic acid and an amine. So next we're going to look at some of these types of reactions as they relate to formation of polymers. So condensation polymers form by condensation reactions. using reactant monomers that have functional groups on two ends of each molecule. So here's an example of something called polyester. So you guys you have polyester clothing or materials, and you've used polyesters very regularly. They're very important materials for people. Polyesters form when carboxylic acid end of one monomer reacts with an alcohol from the end of another monomer, linking the two by an ester linkage. So you can see we lose water and we form an ester. And since both ends of both molecules contain functional groups, you can see that once we've attached one, then the other hydroxide side on the diol can react with another dicarboxylic acid. And that dicarboxylic acid then will have another carboxylic acid functionality to react with the diol. So this can continue onward, and we can make long chains, just like we looked at long chains for polyethylene, which is an addition polymer. This is a condensation reaction, and we can form a polyester from this. We can form polyamides as well. So a polyamide polymer is a type of condensation polymer when we have an amide linkage reacting with monomer units. So the carboxyl end of one monomer reacts with the amine end of the next monomer unit to form an polyamide. So synthetic polyamides are typically called nylon. Nylon is a polyamide. You can see the reaction here. You've got two carboxylic acid ends, two amine ends. One amine reacts with the carboxylic acid to form the amide. And then you've got an amine that can attach to another carboxylic acid, and so on. So this makes a nice long chain. And the long chain forms a molecule which produces our polymer. This also occurs in biological systems where carboxylic acids react with amines, and this is how we form what we call peptide bonds, which are important for formation of proteins. So condensation, polymerization is an important biological reaction or biochemical reaction. The amino acid reacts with the of one end reacts with the amine on the other end. So an amino acid has a carboxylic acid end and an amine end. But in water, because of the acid-base properties of water, typically the carboxylic acid end is naturally deprotonated and the amine end is protonated. This is what's called a Zwitter ion. And we won't get looking at this reaction in biochemical detail in this course. That's left for biological chemistry. But you can see you have a Zwitter ion, and the carboxylic acid end, or the carboxylate end, can react with an adjacent ammonium end, that is the positively charged nitrogen which has the hydrogens. This species here will release water and form our amide linkage. So that's our peptide bond. It's an amide bond. Another important biological system that involves these sorts of bonding motifs are acetals and hemiacetals, which are important in sugars and carbohydrates. A hemiacetal is a particular functional group that has an OR, like an ether, and an OH, like an alcohol. An acetal contains two alkoxy groups, like two ether groups, attached to the same carbon. So in the presence of an acid catalyst, if you have an aldehyde or a ketone and you react it with an alcohol, it produces a hemiacetal. But hemiacetals are unstable, and they'll react with another alcohol very quickly and produce our acetal. Here are example reactions showing how this occurs. So here's the reaction of an aldehyde with an alcohol. It's an addition reaction, right? So this is an addition process where the oxygen from the alcohol attaches to the carbon on the carbonyl, and then the hydrogen atom on the alcohol migrates over to the oxygen on the aldehyde. That produces our hemiacetal. The second step of the reaction is a condensation reaction. So the conversion of the hemiacetal to the acetal is condensation, where this looks similar. We have the alcohol on the hemiacetal reacts with the OH from our alcohol, and this releases H2O to produce our ether linkage, or the acetal linkage. Acetals and hemiacetals can undergo the reverse reaction in the presence of aqueous acids to give the original aldehyde or the original ketone, depending on the system. And the acetal reacts in a hydrolysis reaction to produce the hemiacetal, which then undergoes elimination. So these reactions are all reversible. Make sure you're comfortable with how these reactions move back and forth. So how do we see this happen in biological systems? Well, monosaccharides, that is simple sugars. So a monosaccharide is just a single monomer unit of a sugar that we call, or a polymer that we call saccharide, and we commonly call these sugars. So here's an open chain system for the molecule fructose. And you can see that you've got a carbonyl group and an alcohol group, and the carbonyl group and the alcohol group can unite to form our 5-membered ring, our cyclic hemiacetal. Here's a summary of our functional groups that we've discussed in this chapter. We've got an aldehyde. The carbonyl's got a hydrogen on it. A ketone, where the carbonyl's got 2 alkyl groups. A carboxylic acid, where our hydroxyl group is attached to our carbonyl. An ester, where an alkoxy group is attached to our ester. And finally, an amide, which has NR2 group attached to the carbonyl group. We're going to see, we won't see this in this course, but a lot of biological molecules have multiple functional groups that are present. So in terms of naming, all carbonyl-containing functional groups have higher priority than the functional groups with single bonds to heteroatoms that we discussed earlier. The lower priority functional groups were listed earlier, with hydroxy being the most high, with the highest priority. So here's a quick list of the functional groups and their structures and their names, how we name the systems, and some common examples: alcohols, thiols, ethers, amines, aldehydes, ketones, carboxylic acids, esters, and amides. So kind of reviewing and getting an overview on boiling points. The dominant intermolecular force helps us to predict that. And so hydrogen bonding is the most important, and after that are dipolar forces and then dispersion forces. So here's a list of a series of molecules that have very similar molar masses, and we go from the weakest intermolecular force, which has the lowest boiling point at the top, that is butane, working our way to the bottom. So NN-dimethyl methanamine, here we can see this structure is a tertiary amine, and this has a low boiling point. No hydrogen bonding, just dipolar forces. Methoxyethane, being an ether, has a little bit slightly higher boiling point. The ester, the methylmethanoate, has only dipolar forces, but it has more dipolar forces, and so it's a higher boiling point. The N-methylethanamine is a secondary amine, has some hydrogen bonding, and now the boiling point is increased further. Propanamine is higher still. Interestingly, propanone, which is a ketone, is higher, and it's a little bit out of order seemingly, but the dipolar forces are quite strong because there's a strong dipole moment for this system. And then we can see the alcohol with a single hydrogen bond, the carboxylic acid with 2 hydrogen bonds, and then the secondary amide and the primary amide having substantial hydrogen bonding have the highest boiling points. Now, similarly for solubility, of course, things with hydrogen bonding are going to have the highest water solubility. So amides and carboxylic acids are very water soluble. Things that are moderate to high, alcohols generally are high, amines can be high. Ethers, ketones, esters, and aldehydes are moderate, and then hydrocarbons have low water solubility. That completes our discussion of Chapter 14.